So let's move on. Now it's turn of Ellen Fritsche, and she will introduce us in the mode of action and AOPs. Helen got her PhD at the University of Düsseldorf. Then she spent a 3-year postdoc period at the NIAHS at the Research Triangle Park in the US. And back in Germany, she is now a group leader at the Leibniz Research Institute for Environmental Medicine. So the floor is yours.
Well, thank you very much for the nice introduction, and I would especially like Manuela and EFSA for inviting me to this really stimulating conference here in Milano. And we were actually glad that we could visit the expos. That was a very special event. Now, while waiting for my slides, so talking about the adverse outcome pathway concept, or short the AOP concept, it's really a scientific framework for the management of toxicological knowledge, information, and data. And thereby, it links in a causative way the exposure to a compound to a molecular initiating event, which is a really crucial, the very first event that a compound initiates within a cell or on the extracellular matrix or wherever, and how this is causally causing effects on the organelle, on the cell, on the tissue, the organ, and all the way up to the individual and to population. And thereby, it's most importantly, the key, all these key events, how they are called here, are linked by key event relationship that really needs to be strong to link the initiating event in the very front to the adverse outcome at the end. And thereby, it actually embraces the toxicological pathways and also the mode of action of compounds. And why this concept now taken up by the OECD and very much promoted by them, because it has a certain beauty, because regulators will in the end be able, this is the idea, to use the AOP concept, because if you have a causal link, and not only on the qualitative but also on a quantitative level, then a regulator can actually regulate a compound by its initiating event up here, for example, by using computational tools. That's actually a little bit of science fiction. That's so the future where it's trying to go to, or by using in vitro tests that actually look at the effects here on the organelle or on the cellular level. But by creating thresholds and by using the AOP in a very stringent way, regulators will be able to use this concept. On the other hand side, it's actually an opportunity, like a crossroad for academia and industrial researchers to interlink with regulators, because academia researchers do, I mean, this is what we do, this is our everyday business, to look what are the molecular effects on cells and organs and tissue. And by knowing about the AOP concept, academia can also contribute and produce research that in the end regulators can use. And because the communication between regulation and academia is really important, I think this concept is beautiful for actually doing this. So now in my talk, I will first give you a hypothetical example. Why do we need something like the AOP concept? Then in the second part, give you actually an example of my own laboratory, and in the third one give you an example from EFSA, how EFSA is actually right now using this concept. Now, the first thing is, why do we need this AOP concept? For example, to evaluate the theory of storks, which means babies are brought by storks. And in 1988, Helmut Ziesis, actually from my university in Düsseldorf, published a paper in Nature where he showed that there is a very nice correlation with the decline in birth rate in West Germany, which was really threatening at that time, and actually the breeding pairs of storks in the area. So he said there is concern in West Germany over the falling birth rate. The accompanying graph might suggest a solution that every child knows makes sense. So how can we test now if such an hypothesis holds through? Well, if you calculate, so it would be 500 babies per year per stork pair. That makes sense. If a stork pair brings 2 babies a day, can take vacation in the wintertime. Well, there is another a graph actually concerning pesticides, and that was the level of DDT that was found in breast milk in Swedish women. And this looked very similar actually to the graphs here of the declining birth rate and the number of stork pairs. So the hypothesis would be, okay, the declining DDT exposure, and this is totally hypothetical, so this has no scientific impact to it. So the falling DDT exposure decreases the stork population, which leads to a decrease in babies in West Germany. So you can use the AOP concept to test such an hypothesis. So I did a PubMed search for DDT and storks, and I actually retrieved one publication showing that in India there's actually DDT found in an Indian stork population. However, looking a little bit closer into the literature, what you all know, DDT is an endocrine disruptor, causes eggshell thinning in bird populations, and actually leads to bird population decline. So there was no indication that our first link here could be causally linked. So there's no literature support that DDT exposure could lead to a decline in stork population. However, and storks have really rarely been seen at sites of baby delivery. So this is actually making the second causal relationship, invalid. So this is kind of an easy way showing why do we need an AOP concept and how can this work. Now, I will come to an example from our laboratory, and this is actually real science, not like the stork theory. It's the developmental effects of high-dose plant extracts. And this is about food supplements which really contain high amount of plant extracts or even their pure ingredients that are freely sold through the internet, for example, and freely available. General toxicities are usually known for these compounds, but consumers, they consider these herbal-based food supplements as healthy and safe, and also there's evidence now that young women increasingly use them during pregnancy because they think, they're improving the health of their babies. One of these compounds is the green tea catechin epigallocatechin gallate, or short EGCG, and you find this everywhere now, coming from a supplement to Coca-Cola or Kit Kat or some other sweets and candies, and you can also buy it at really high doses because they have potential health benefits in preventing or treating diseases, including cancer. The proposed mode of action is that EGCG interferes with cell proliferation, adhesion, and migration. And actually, these processes are also essential for brain development. Well, this is why it's really disturbing that there are actually preclinical studies to prevent developmental fetal alcohol syndromes at an adverse effect with a compound where actually the potential to cause neurodevelopmental toxicity is not known at the moment. And this was the reason why we actually tested this compound in what's called the neurosphere assay. It's similar to what Thomas showed you with the mini brains. There are three-dimensional clusters of neuroprogenitor cells, but which don't stay as a brain ball, but actually the cells migrate out here. And this compound, EGCG, inhibits the migration of rat and human neuroprogenitor cells, and humans are more sensitive than rat here. And when you look at the pictures here, you see that there are these little arbors. When you compare this to a normal migrated neurosphere, you can immediately see that this is not really a normal neurosphere phenotype that we observed. So putting this into the AOP concept, because this is what I'm trying to do as a molecular scientist right now, so that regulators may later use my data. The cellular effect, so we're in the middle of the AOP, so we are trying to develop it from inside out, is disturbed neuroprogenitor cell migration is caused by epigallocatechin gallate. Now, how does it work? Well, others have shown by affinity chromatography that EGCG can actually bind to laminin, which is the predominant extracellular matrix protein in the human brain. And so we took this data, and here M is standing for matrix, C is standing for cells. If we treat the matrix and the cells, or only the cells, or only the matrix, it's very obvious that EGCG here is producing these phenotypes only if the matrix is actually exposed. An increase in amounts of laminin actually antagonize the inhibition of migration by EGCG, adding to the AOP concept that actually the EGCG is inhibiting migration through interaction with the extracellular matrix by binding to laminin. Now, migration is actually consisting of motility and adhesion. So a cell has to be able to wander, but also to adhere to the extracellular matrix. And therefore, we actually looked at our neurospheres and looked at the motility. And you can see that all the cells, they are very motile, they are migrating out nicely. But then, what we have never seen before, they move back in because they cannot really adhere, and when the cells can't adhere, they think they're in the wrong place. So even in an in vitro assay, you can actually assess that something strange like this. The cells move out and they move back in. And this is what you don't see when you just look after 24 hours, then you don't see any cell outside, but this is what's happening in between. So now that we know it's not motility, it's adhesion, it has to do with laminin. Laminin binding of neuroprogenitor cells is actually mediated through extracellular receptors. They're called integrins. And this laminin integrin binding is transported by integrin beta 1 or beta 4 containing subunits. And by using an antibody against the beta 1 or 4 integrin, so blocking their ability to actually bind to the laminin, we see down here is the picture of the beta 1 antibody, a very similar phenotype than treating the cells with EGCG. And when we use the beta 4 antibody here, we do not see this at all. And we do not see this with compounds which disturbed actually motility of the cells. So our inhibition of migration is actually, it's working through the beta 1 integrin. So how can we now get, we also did actually an in vivo study because we wanted to know, so is this something that is really transported to the organ level with the EGCG, because an in vitro method as a standalone you cannot really use. And what we saw is that when we treated pregnant animals with this compound, we got a lower density of BRDU positive cells in cortical layers, indicating that there were less cells actually reaching the cortical layers. How can we link this now to this beta 1 integrin, to the mode of action? And there's actually data from transgenic beta 1 integrin deficient animals, which also show that they have lower density of BRDU cells in cortical layers. And this is because the glial cell alignment and adhesion is actually disturbed in these animals. So we're trying to fill what we find in the lab with more literature and more supportive data. And actually these animals with the beta 1 integrin knockout, they also show behavioral deficits. Now, trying to find this disturbed glial alignment, we thought, well, we have actually a perfect model to do so, because the neurospheres, the glial cells are growing out radially from the sphere core. And this is actually this is a schematic view here of a migrated neurosphere, and we measured the angles with the control and the EGCG treated cells, and measured in what angle do they come out of the sphere. And these polar plots down here indicate nicely that in the controls, the majority is between plus and minus 30 degrees, and the more compound you get, there is more deviation in the angles, meaning that the cells, they can't adhere, and so they are going all over the place. So what they saw in the beta 1 integrin knockout animals is something similar that we even can reproduce here in vitro. And this actually causes a reduction, a secondary reduction of neuronal cells here in these cultures, so that we can add another error here. And so this is a very preliminary AOP, a very putative AOP, and what you really need for filling it up is more data, of course, but also what you need is to integrate exposure. So at what levels do all these effects occur to get from a putative, qualitative, actually to a quantitative AOP. And this is really a big issue at the moment in developing AOPs. And to make a long story here short, we went through a hypothetical pharmacokinetics study, what's known from actually EGCG exposure in humans, extrapolating from rats to fetal brains, and we came up that when you take a 3-gram dose with 2 spoons of these high concentrated EGCG dose, you will end up with about 1 to 3 micromolar in actually human fetal brain. So this is just by doing a literature research on the kinetics. And actually our LOX for the human neuroprogenitor cells are in a very similar range. So this AOP is a new mechanism to have a concern for compounds which interfere actually with this extracellular matrix binding during development. Now, very briefly, coming to the end, coming from actually this is EFSA work, because this is an assessment of paraquat from the US EPA stating paraquat is not a neurotoxicity hazard. And EFSA has recently re-evaluated actually exposure to pesticides and health effects. So, and this is published here in this external scientific report. And they have shown that pesticides used is through epidemiological studies linking the exposure to Parkinson's disease. And the animal studies are controversial. It's not really clear if they are causing now Parkinson's disease and actually also childhood leukemia or not. And this is why EFSA has actually formed a working group for the re-evaluation of. Oh, great, so I'm good in time. So this is my last slide, actually, for doing a real evaluation of for assessing risk factors for Parkinson's disease and also childhood leukemia by using actually the principles here established for the adverse outcome pathways. And I think this is the first time that EFSA is using the AOP concept for really evaluating a compound. It's different pesticides, not only one compound, but for reassessing really human risk, because the animal experiment, what Thomas said earlier, sometimes is just not good enough. And this covers, of course, all what I've shown you before, mechanisms of action. It's doing good literature studies and trying to set up such an AOP for these two diseases, childhood leukemia and Parkinson's disease. And yeah, I hope I gave you a good overview of how to use an AOP, not so much about the guidelines, how to build one, but yeah, it can be used for coming from the epidemiological side, coming from the lab side, and like I said, I think it's a really good interface for research in the lab and regulators. And this, I would like to thank my group who worked on the EGCG project, and I'd like to thank you for your attention.
Many thanks, Helen, for this brilliant presentation. I like your example on food supplement because usually people think that they are safe and healthy, and it is not the case in most of the time. Are there any burning questions for Helen? Yeah, we have one there.
Hi, can you hear me okay? No. Can you hear me now? Better? Yeah. I'm Helen Clegg. I'm the toxicologist from Mars. So just the company, not the planet, just in case there are questions. I guess what I was coming to was about the green tea and the EGCG, because obviously that's got issues with liver toxicity as well. And I know several countries within the European Union have withdrawn it as a dietary supplement because of issues there. So I guess with the AOPs, it's how do you ensure that you are picking the right endpoint for the overall risk assessment? So obviously here you've pointed out a developmental issue, but actually if that's not going to be the pivotal issue that drives the risk assessment, there's a question there. So I don't know, how do you make that decision about which AOP is driving a risk assessment?
Yeah, I mean, you need many different AOPs, and we are just at the beginning. So you basically need an AOP for every single organ, because every cell has its specialty in toxicological thresholds and in signaling thresholds. And you need a testing battery of in vitro tests, I mean, to see, so where is my most sensitive endpoint? So this could be part of an IATA, for example. And it's, I mean, this is true for different organs, but this is also true for different endpoints within one organ. So it's you need to find with such integrated testing, or maybe in the future with computer modeling, where your most sensitive issue really lies, in which organ, on which endpoint. And therefore you need a bunch of AOPs. And one AOP is far not enough for anything anyhow. So there will be AOP networks built, and there will be AOPs spanning through all the different questions that you have. So this is really in its infancies right now. But yes, no, with just one AOP you can't.
Thank you. I will suggest to take your question for the panel discussion, and we have to move towards the